Good morning, good afternoon, and good evening from wherever you are calling in. I want to thank LabRoots for providing the opportunity for us to speak today. My name is Jeroen Aerts, and I'm a customer technology advisor at Resolve Biosciences. And at Resolve Biosciences, we are developing molecular cartography, which is a highly sensitive and highly multiplexed spatial transcriptomics platform. And I will be talking more about how we can use our platform really to empower scientists to discover the next breakthroughs in their research. And more specifically, I will be talking about that in the context of neuroscience, how to use our technology to study gene expression changes in the brain with spatial context. And you see behind the logo already a little teaser of what it actually looks like. It looks like a piece of art, and I think it's absolutely beautiful. But what it actually is a cross-section of the mouse hippocampus, and many of the colors you see are actually individual molecules, individual transcripts that are colored based on the genes where they reside from. So just to dive in deeper on why spatial technology is actually very important and is considered the next frontier in our understanding of how tissues are composed and how we understand the cellular heterogeneity. So over the past couple of years, as you may know, we really increased our understanding about the heterogeneity of different cell types in tissue through the revolution of single-cell RNA-seq and other technologies. And how that actually basically works is if you have a tissue, in this case, for example, a mouse brain, you need to dissociate that tissue. So you need to extract all the individual cells and you compartmentalize every individual cell, be it in a droplet or in a microplate. And within each droplet or well of a microplate, you will generate cDNA, generate a library for sequencing, and this will allow you to really profile the transcriptome of each individual cell. And it's a very, very powerful technology that, coming from, let's say, bulk RNA-seq, really allowed us to understand the cellular heterogeneity much better and really allowed for many new discoveries in rare cell types and how they respond to pathology or drugs, etc. But of course, when dissociating your tissue, it's difficult to know how these cells relate to each other in a spatial context. You might know where approximately they are residing. So in this figure, you can see maybe the red cluster, these are probably cells originating from the cortex in this tissue, but it's difficult really to use that as an extra dimension. So I think spatial technology in this sense is really the next frontier, the next dimension we need to really understand those things. So, and how it actually works is you take also in this case, for example, a mouse brain, and you make a section, and you will directly in that section in situ target those same transcripts and really visualize them within their native contexts. And what you are allowed to see then is let's say the same clusters, the same populations of cells that you would normally identify with single-cell RNA-seq. You can actually look at them in a spatial context, how they relate to each other. For example, in this case, how they are organized across the mouse cortex in different layers. And this adds a whole new dimension on how we understand the underlying biology. And within the neuroscientific context, of course, as posed by Ramon Cajal almost a century ago, this will also allow us really to understand much better the cell-to-cell communication, which is fundamental to understanding the different networks and how all these neurons and other cell types communicate with each other with very high resolution while maintaining a spatial dimension here. So I want to walk you through, apologies. So before moving into some examples, I want to make sure that you understand the importance of the spatial biology in this context. And it's really also acknowledged by Nature Methods because they announced that spatially resolved transcriptomics is the method of the year 2020. And many of these technologies really are being developed as we speak, and many companies are in this space. And we at Resolve Biosciences truly believe that our very sensitive and highly multiplexed platform can really empower scientists to discover new things and boost their research. So to dive deeper in what is possible with our molecular cartography technology, so this is a cross-section, a coronal section of a mouse brain. So this is taken from the Allen Brain Atlas, and on the left side you can see the bright field image, and on the right side is an annotation from the Allen Brain Atlas depicting the different areas of the brain, and really to localize that, let's say, in an anatomical atlas. So what we actually did is we used our technology to profile up to 100 genes in one sample simultaneously, and we were able to capture more than 8.5 million individual molecules. So superimposed on this image, you can see the visualization of our data set with all 8.5 million molecules visualized here. So if you would in fact zoom in, and I will show you images later on, you can really pinpoint each individual molecule separately, and this provides a lot of opportunities to really dive deeper into this data set and understand how the different transcripts, but also the different cell types are related to each other. So how did we actually do this? Well, our molecular cartography is actually based on a combinatorial single molecule FISH approach. So let's say you have a tissue section, in this case, again, our mouse tissue section. We develop transcript-specific probes for a list of genes that you can provide, so you can fully customize that. And we hybridize that tissue with all the transcript-specific probes. And we have a proprietary chemistry where we are able to color these transcript-specific probes, but also decolor them. And we use that in a sort of cyclical or repeated imaging cycle. And why do we need repeated imaging cycles for this? Because this actually allows us to, with high confidence, trace back the location of each transcript and annotate that transcript to a given gene. And so on the right side, you will see an example of this, an illustration. Let's say we have 6 imaging rounds. And in each imaging round, we know beforehand how we designed our probes, what color we would expect in each round for a given transcript that originates from a given gene. So for example, transcript coming from gene A, we know upfront that it would be fluorescent in the green channel, then again in the second round in the green channel, again in round 3 the red channel, etc., all the way up until round 6. And at the very end, we can just decode that barcode basically and really annotate specifically, as you can see here highlighted in orange, where those transcripts are. We can, with high confidence, identify them as being originating from gene A in this example. So it's both very specific, but also highly sensitive, because the sensitivity comes from the fact that it's based on single molecule FISH. And I will talk a bit later on that today as well. But first, maybe more about the specificity. So we compared this data set with older publications using traditional in situ hybridization. So on the top panel, you see many different marker genes for cortical layers in the mouse cortex. So we were able to nicely recapitulate that with our technology. So on the bottom panel, you see for these individual genes, very similar expression patterns, and even for example, CTGF on the right side, we can actually see very clearly that this is a specific marker for layer 6B in the mouse cortex. But the big difference here is that the data set shown on the low part, so for each gene, we just colored this in green, right? But basically, each of these panels are comprising of a huge data set where we actually multiplexed up to 100 genes. So that data set is much richer, and we just pick out the gene we want to just visualize that and do further analysis. So that adds more depth to the data set we have compared to, of course, low multiplexed in situ hybridization. And as for sensitivity, so when we compare that with bulk RNA-seq coming from the Human Protein Atlas RNA-seq database for mouse brain, we can nicely see that there's a good correlation with the counts we have from the molecular cartography. So genes that are expressed highly in the RNA-seq data set will also be highly expressed in our data set, and vice versa for low expressed genes. But I think more importantly, we want to, of course, compare that to another spatial technology, and in this case, single molecule FISH, because it's considered to be the gold standard and the most sensitive spatial technology available to date. So we compared our technology with single molecule FISH just for a couple of genes, and we see that the counts per cell are almost identical. So we have similar sensitivity as compared to single molecule FISH. So let's dive deeper into what we can learn from data sets in neuroscience. So again, this is the mouse brain hemisphere, and within the set of 100 genes that we selected, we just visualize here 13 of those genes, and these 13 genes are actually markers for the different cortical layers. And zoomed in centrally in the slide, you can clearly see that as we expected, all these different markers, it's a beautiful rainbow picture here, but it nicely shows that we are able to confidently recapitulate what is known to be specific for the different cell layers, and that these marker genes are nicely located to where we expect them to be. So that's a nice validation of the marker genes we put in, and the validation of our data set as well. But you can go further. So what is nice is within the same data set, you can actually really dive deeper into all the different cell types. So you can clearly see here that in the different shades of red, we visualize the glutamatergic neurons, but also in yellow, the GABAergic interneurons, and also microglia, astrocytes, oligodendrocytes. So these are all nicely visible in this data set. Of course, it is a lot. The data is highly dense, but it also shows nicely how many individual data points you're able to capture just from 35 genes, so not even the whole data set. But you can still clearly see already, for example, in yellow, these GABAergic interneurons nicely interspersed with the glutamatergic neurons beside it. But of course, I think it's nice to dive even deeper into the data and really it allows you to just select a subset of genes you're interested in, and this is a great example of it. So if we only look at certain subtypes of inhibitory neurons, when we just look at the marker genes for that, for example, we have GAT1 or parvalbumin positive interneurons, somatostatin in yellow, and VIP interneurons in purple. You can nicely appreciate, I think, the complexity and the way they're organized, and even especially for the somatostatin interneurons here, you can even see the projections coming out of the cell body here, which is a nice thing to see if you're interested in neuroscience, to also see all those details popping up. And importantly here, I want to show you a visual representation of one single somatostatin interneuron. And this is really to show that we are, for one, able to capture 3D information in our data sets. And this is nicely shown by the DAPI segmented nuclei. And you see each individual spot represents one individual transcript molecule coming from somatostatin. You can nicely appreciate that even zooming into one individual cell like this, it allows you to really dive deeper into subcellular differences, either within a given cell or comparing that to other cell types as well. So our resolution is really subcellular down to the single molecule level. So More downstream analysis, of course, can be done. And one way to do that is, of course, taking that whole data set and segment out each individual cell. Of course, segmentation is a topic in itself. It's a very complex topic, especially in neuroscience, because all these cells have very complex shapes. But that's a topic, I think, for another webinar. Today, I want to show you, if we just take basic cell segmentation based on DAPI, we're already seeing a lot of information popping up. We can generate cell-to-gene and gene-to-cell matrices. And what you can see is per individual segmented cell here, we are able to pick up hundreds to sometimes thousands of individual signals per cell, and also upwards to 60, sometimes more genes per cell that are actually expressed. And this is in a panel of 100 genes. So that tells you already a lot of the intricate details of the data set and how much information you actually capture. But using that cell segmentation, what it allows you to do is really, just like in single-cell RNA-seq, it allows you to take that information, take that matrix, and do again clustering on that. So it's completely compatible with, for example, SERAT. And in this example on the left panel, you can see over 35,000 individual cells based on the 100 genes data set I showed you earlier. And you can see that all these clusters are very nicely defined, and we were able to identify all the major cell types in this. And on the right side, you can see the heat map, and this will allow you to identify different marker genes that are specific for each individual cluster. But of course, very different from single-cell RNA-seq is we have here quite familiar looking data, I think, but what is important here is that we always retain the spatial dimension here and the spatial structure. So what is shown here is you can always go back and, for example, select a certain cluster that you're interested in and see how it actually maps spatially in the section where you captured the data originally from. So here is an animation that actually shows all these different clusters being projected back onto the tissue. And this really allows you to understand much better how all these individual cells and these different cell clusters are spatially mapped. And this really provides the spatial context whenever it's needed. You can always go back to that again. And this is also represented here in the slide where we are nicely able to visually represent all the different cell types on the tissue section, whether it be oligodendrocytes or, again, the glutamatergic neurons or microglia. You can really appreciate all the different spatial organizations of these cell types. And what you can do, because we have the segmented cell segmented data from that, you can actually just use that clustering and count all those different cells and see, for example, in a simple analysis, how many glutamatergic neurons are there compared to other cell types. And we did this on our data, and it really nicely recapitulates what is known in the mouse brain already. So that's, again, a nice validation of our technology and what you can do with it. And this really opens up the whole new spatial dimension in your single cell data set. But I want to talk also today more about certain applications, what you can do. So for example, what we did here, I want to explain a bit more about what we did in a collaboration with a major pharmaceutical company, and this was in the context of Alzheimer's disease. So in Alzheimer's disease mouse models, what we know from the past couple of years based on single-cell RNA-seq is that, of course, in Alzheimer's disease, you have the occurrence of these amyloid plaques, which is a typical phenomenon of the pathology there. But what was discovered is that you have a specific subset of microglia that respond to this pathology, and they're called disease-associated microglia. And you can identify these subtypes using single-cell RNA-seq. But I think it's very important, and it's a very nice use case of the power of spatial biology to really understand how these subtypes of microglia are responding to the pathology where they are located, and this can provide a basis for drug discovery and really modulating the outcome of disease. And just to give you more insight into this, so on the left you can see comparison of gene expression between wild type and an Alzheimer's disease mouse model, and on the top you see just the depiction of the different cell types, neurons and microglia as well, microglia in green, neurons in blue here. And on the bottom, you can see that when coloring the same cell populations, you can immediately see big differences. And we see less neurons there in the tissue, but also slight increase in the green signals, meaning the microglia. But importantly, I want to mention here as well, in yellow, you can see here the amyloid plaques. And what was actually done is we first ran our molecular cartography workflow. And after that, because during the workflow, we have the tissue is completely intact. We maintain the tissue integrity. You can actually, after that analysis, use it still for immunohistochemistry or labeling it with a dye. And in this case, we labeled it with a PFTAA dye, which specifically binds to the amyloid plaques, here depicted in yellow. And on the right side, this is another type of analysis. We can just visualize just all the genes that are enriched in the wild type cortex versus the Alzheimer mouse model cortex. And we can see that there are major differences, of course, and many of these genes are actually enriched near the amyloid plaques. And again, when we segment out all these cells using DAPI with QPOP, again, like mentioned before, we can generate these UMAP clusters and really identify all the different cell populations. And just like you would do with single cell RNA-seq, is you can compare wild type versus AD mouse model clusters specifically, and you can compare, let's say, the effect of pathology in your transcriptomic readout here. And there are many different differences across many different populations. But what the spatial component allows you to do, and I will go into detail a bit further, but it allows you to also measure the distance towards the plaque center for each individual cell in this cluster. And this is just, say, a heat map, and we can see that for microglia, for example, so the top cluster, you can see that you have two populations. One is related to the wild type, the other one is related to the AD pathology. You can clearly see that it's also heavily correlated with the distance towards the plaque. So the closer you go to the plaque, the more you see these cells going into a disease-associated phenotype. And you can really dive much deeper into this analysis. And when we zoom out a bit again, and we just analyze all of the frequency of all the different cell types, we can clearly see that when you compare wild type to AD mouse model brain, you can see a decrease in glutamatergic neurons and an increase in the number of microglia. But of course, what is important is you have the spatial component at all times. What we did here is we actually correlated that, or quantified actually the distance towards the closest plaque for all these different cell types. And on the right panel, you can clearly see that when you go closer to the plaque center, you can see a decrease in the amount of glutamatergic neurons, and you can see an increase in the number of microglia. This already hints towards what is happening in response to pathology. You can even go deeper into the data and really check individual transcript levels. So what we actually did is we analyzed more than 600 individual plaques across 3 different animals, and we drew concentric circles around the plaque, and then we measured for each transcript how their levels changed in relation to the distance to the plaque. So, and this allows you also on individual transcript level to really assess potential markers for different cell types. So in this case, this is visualized for a marker gene that is known to be a marker for the disease-associated microglia. You can clearly see in the middle panel that they're located around the amyloid plaques. And on the right panel is a quantification of this that you can see for two markers. So DAM marker 1 and 2 in green and red, you can see an increase in their expression the closer you get to the plaque center. And at the same time, for some neuronal markers, you actually see a decrease. And this is a nice readout to have if you're, for example, developing a drug and when a modulated disease, you would also like to see changes in these transcripts, but also on cell type level and things like that. So it's, I think, a very nice application and shows the power of spatial biology in the context of new discoveries in Alzheimer's disease. And again, so the previous slide was more on the transcript level, but you can always zoom out and really look again at the different cell types and how they respond to pathology. So on the left side is again a data set on the AD mouse model, and on the right side you have the wild types. And if you just map back the different cell types, in this case, for example, you can nicely see on the right with the wild types in purple the astrocytes that are nicely distributed along the mouse cortex, and the reactive astrocytes that are on the outer edges of the cortex. You clearly see in the mouse model representative for Alzheimer's disease on the left side that these reactive astrocytes are now dispersed throughout the cortex. And again, these disease-associated microglia, they pop up as a new population and they surround the amyloid plaques, again labeled with the PFTAA dye. So you can learn a lot about these different cell clusters, how they map back spatially, and how they interact with the pathology in this case. And this is also an aim. This is an ongoing work, just to show you that it's possible to also do this on human samples. So this is again in collaboration with a pharmaceutical company to really show differences between control human brain tissue and post-mortem Alzheimer tissue in Braak stage 6. And you can see here just a snapshot, a teaser basically of the different cell populations we're able to visualize here. And you already see clear differences in for example, the expression of neuronal markers here. But this analysis is ongoing, and I hope maybe in the next webinar dive deeper in the outcome here and what you can discover with our technology on human brain as well. This shows you that it's that our technology works in different species on different sample types, etc. And I want to summarize what is possible with our technology. Basically, we are able to apply our technology to many different species, to many different types of tissues, and also different cell cultures and cell types. And we can probe up to 100 genes simultaneously in each individual sample. What I think is very important is that we offer a very high resolution, so it's single molecule, and we are also able to provide 3D information across the tissue section. As for analysis, which is a very important part of the whole spatial technology field at this moment, we actually provide an interactive software to really allow you to analyze these images very intuitively, but also it's compatible with open source platforms like I showed you, like Seurat and things like that, to really allow for many different types of analysis. And I think very importantly, one spot is one transcript. So it really allows you to quantify our data digitally. So we don't rely on, let's say, fluorescent intensity. So this is really a nice way to quantify your transcriptomic data in a spatial fashion. We also offer the possibility to do a multi-omic readout. As I mentioned throughout our assay, we make sure that the tissue remains intact. So you can do afterwards, you can do immunohistochemistry or combine it with dyes, with other types of readouts. And the throughput in the system we are developing in our workflow is actually 24 samples run in parallel for 100 genes per sample. And I think our sensitivity, as I showed before, is very on par with single molecule FISH. And this is also partly because we don't use enzymes and we don't have like an amplification bias inserted in our chemistry. And this is one of the differentiators of Resolve Biosciences, we actually offer the high resolution and the high sensitivity. And this is not always the case. For example, when you look at other spatial technologies that use rolling circle amplification, or when you have technologies where you need to extract the RNA with permeabilization onto a glass slide for sequencing and things like that. So we offer both a high sensitivity and a high resolution. And we maintain our tissue integrity, of course, which allows you to do much of the downstream analysis. And I want to end with just an announcement, like how we work today is we offer an end-to-end service and support. So you are able to access our technology in service mode. So if you have a gene list available, you can actually order probes with an online tool. We can support you in that. What we actually do is we send our special Resolve slides to you. You section your tissue, you put the sections on top of our glass slide, you ship the glass slide back, and we run the chemistry in-house, and we provide you with all the data you need to analyze and interpret your important biology. And we are there really to support you in producing the best biology and really to boost your research. And I want to end with the last slide. If you're interested in our technology, please drop me an email if you want to make use of our end-to-end service. Again, don't hesitate to contact me. And if you have any questions, you can also email. But of course, we have a Q&A lined up as well. So feel free to drop a line there. So thank you very much and have a great day.